All right, hello and welcome to our final STEM Talks of the year. Thank you to all of you that have been with us in September, but if you haven't, this is your first event, we're very excited to have you here. So our very last career panel for this academic year is on vision sciences, and you will learn about what that means, but hopefully you can kind of guess from the pictures. We're going to be focusing about the eye. So if you've been here before, you know how these events work. But basically, this is a career panel, and we have three amazing individuals that will talk about what their career entails. We'll talk about what they do on a day-to-day basis, how they got to where they are, and then we'll have time to answer your questions at the end. So feel free to put your questions in the chat, or you can also raise your hand at the very end of the presentation part of the talk. As you can see, though, the event is indeed being recorded, so please just try to keep your microphones and cameras off to not distract from the recording. And then if you are here for the certificate program, as long as your first and last name is spelled properly, you are good to go. So before we formally start the panel, we're just going to do a little bit of a welcome poll. Trisha, can you launch the welcome poll? We just want to know if you've been to one of our events before, and if you are interested in the vision, eyes, anything in that field. So we'll leave this question up for about 30 or so seconds. But for the sake of time, we're just going to start with our panel. So Kristen, whenever you are ready, feel free to take over screen share and talk about your graduate work.
Okay, so hi everyone. My name is Kristen, and I am a Master's of Science student at the University of Toronto with the Institute of Medical Science. And I am in the second year of my Master's and hoping to hopefully transfer into my PhD this summer. I work in the lab of Dr. Brian Ballios, who is working at the Krembil Research Institute connected to Toronto Western Hospital. And of course, in theme with today's presentation, I am in the vision science research program. So I do all vision related research, mostly pertaining to the retina and retinal disease. So a little bit about me quickly. I'm originally from Oakville, and I went to the University of Guelph for my undergrad, which I'll talk about in a little bit. And now I live in downtown Toronto and commute to my lab every day. And some fun facts, I have two golden retrievers named Chelsea and Jasper, who are the loves of my life. And I really like traveling and enjoying some very good eats, which is very helpful in Toronto because there's lots of fun restaurants. And I love to cross-country run outside, particularly in the fall, which is where this photo on the right was taken in Webster's Falls in Hamilton. But city running is also fun too. So in terms of my education, I went to high school in Oakville, graduated in 2018. Then I did my undergrad in biomedical science at the University of Guelph, which is an excellent program. And for those of you who are not graduating this year and are in grade 11 and are considering something in the medical sciences for your undergrad, I highly recommend that you consider that program. It was excellent. And in terms of graduate school, now I'm here at the University of Toronto with my Master's of Science underway and hoping to do the PhD soon. Then after that, hoping to do medical school and combine the PhD and MD aspects of my education in a clinician scientist career position eventually, many years down the line. So I'll talk very briefly about my research. I'll give you like the overarching view of it, and then in the question period, if you have more questions, I will be happy to answer them. But basically, I work to study rare genetic eye diseases that do not have cures yet. So being genetic eye diseases, they're caused by mutations in specific genes that affect the functioning of the human retina. The retina is that area of the eye at the very back. It's a thin tissue that has many layers of cells. And the photoreceptors, which are at the very back, the final layer of the retina, that's where light's transduced. And when you have certain mutations in certain genes, those photoreceptors degenerate over time in those patients. And because of that, patients with these inherited retinal diseases experience progressive vision loss and eventually blindness. And because we haven't been able to fully understand the mechanisms for which these diseases occur, we don't yet have a cure for most of these inherited retinal diseases. And that's what I'm working to help take a small step in developing using stem cells and stem cell therapy. For those of you who are not familiar, stem cells are blank slate cells with a very magical property of being able to differentiate into all different cell types or make more stem cells and regenerate in that way. And how they can be applied in a clinical context is basically by implanting healthy donor stem cells into a diseased retina and allowing them to repopulate a portion of the retina to restore function of cells in that area and hopefully restore vision. So the model I use to study the cell therapy and also studied specific inherited retinal diseases and their way of manifesting is using retinal organoids. Retinal organoids are these 3D tiny little retinas that we grow in a petri dish, and we actually use patient cells and reprogram them to stem cell state from a blood cell culture. And we allow the stem cells to form and form these 3D structures and all these different cell types that form this retinal tissue. It's like about this big, it's like a grain of salt. And it has all the different cell types spontaneously formed into like a laminated structure called a retinal organoid. So when we use cells extracted from a patient that has a blinding eye disease, then we can expect that the retinal organoid that we generate in the petri dish derived from those patient cells will manifest the disease in the little organoid that we're studying. So because of this, we're able to study the ways that the disease manifests without having to use a retina from a human, which we can't do live. So it's a humanized model without having to use patients themselves, which is really cool. So these figures I threw up onto the slide are to visually show you what that's all about, but I won't get into it because it's a little bit complicated, but you just see on I think the most important one is probably those that bottom panel with the red and the green little balls, which show you what organoids look like at a mature stage. The healthy ones are the ones that are in red, and you can see that they've formed this nice layering with this little hair-like projections at the end. Those are the photoreceptors. So they're forming well in the healthy organoid from healthy cells. And then organoids that are derived from patients with diseases, you can see those little hair-like projections that are coming from the border of that ball are much shorter and much less robust. And that's because they have the disease. So I'm just going to talk a little bit now about my week in the life and what it looks like to be a master's student in a wet lab. So there isn't really it's not like a 9-to-5 job in the corporate world, grad school, where you rise and shine at a specific time and you have your schedule for the day and then you go home at 5 p.m. It's a lot, I guess you could structure it like that if you wanted to, but sometimes experiments go for a super long time and it's not set in stone how long you're going to be in the lab. So sometimes you get there really early, sometimes you get there late, and then you end up staying until 8 p.m. It really depends. Some experiments take 4 to 8 hours to do with lots of incubation times. So you're basically determining your schedule based on how your experiments unfold in the day. And outside of experiments, other things that fill my time as a grad student are reading papers so that I become more knowledgeable in the field that I'm researching in, and going to classes just like you would in undergrad. We have to take a few classes as master's students to gain background knowledge in our field, and lots of lab meetings. And let's see if I can play this video of me at our cell culture hood doing some work there. Okay, so you get the idea. And then pause this, I can probably go forward. Here are some other aspects of life as a grad student. Another great thing is you get to travel a lot, which is amazing. I just came back 2 weeks ago from Italy at a conference, and before that, Seattle. And then these are some older pictures from last year. So it's a really great opportunity to present your work and research to other scientists in the field by traveling to these conferences. And then, of course, city living in Toronto is always fun to show off. So there's a picture of Chelsea at a park in Toronto on the right. And then in terms of the rewarding aspect of being a grad student, to know that you're playing a small part in a bigger goal of whether it's helping to try and develop a cure for a disease or contributing new knowledge to a field, it's rewarding in itself to just play a small part in that. And if you're able to help people and have people count on you and know that you're trying to improve their life in some way. So for me, with these patients that don't have a cure yet for a blinding eye disease, that motivates me every day to get up and try to play a small part in hopefully developing that cure one day for those that need it. And one day hoping to bring that research knowledge into the medical field, that's the ultimate goal. And I think that's it for me for now. And then I'll come back for the questions at the end.
Perfect. Thank you so much, Kristen. So many cool things happening in your work. All right, Tahani, you are up next, so feel free to take over screen share when you're ready.
Hey, good evening, everyone. My name is Tahani Baakdhah. I'm a postdoc researcher currently working at Dr. John Vac Lab in the Donald K. Johnson Eye Institute. I am So a little bit background. So in addition to being a scientist, I'm also like doing lab work since a long time, trying to discover new things in my field. I'm also a science communicator, so I like to use art, especially crochet and knitting, to create educational models so that I can use them in explaining my research and other people also to other people like complex concepts of science, so that we can make it more easy to understand and fun to learn as well. Little bit my background. I graduated with a medical degree back in 2003 from the Faculty of Medicine in King Abdulaziz University in Jeddah, Saudi Arabia. Afterward, I did post-medical training for 3 years in Jeddah too, then moved to Canada, where I did a master in molecular genetics, and then moved to Toronto, where I got my PhD from University of Toronto back in 2001. And currently I'm working as a postdoc, 4th-year postdoc. So during my PhD, I was interested, because one of my hobbies was photography, really. So I used to go outside, capture those beautiful scenes, looking at all those colors, and I was always fascinated by how can we see these beautiful things around us. So during my PhD, I was looking for a vision science program to understand deeply how our eyes work. And I was really lucky to get a PhD at Dr. Van der Kooi Lab in the University of Toronto, where we were using mouse and human models to study more photoreceptors and pigmented cells in the eye, and how they connect, how they work, how they come out of stem cells. So those stem cells, like every one of us, if you see the dark line surrounding your iris, there is like 1 in 500 of those cells are stem cells. Those stem cells produce like daughter cells that can differentiate into photoreceptors and to other cell types in the retina. As Kristen explained to you, that retina is the back layer of the eye where all those photosensitive cells reside. So those stem cells later differentiate into photoreceptors and the pigmented cells in the eye. And that was like my goal during my PhD, is enriching for those stem cells and their daughter cells to make more of those photoreceptors and more pigmented cells in the eye, so that we can use those cells to replace lost cells in patients suffering from degenerative eye disease like macular degeneration, retinitis pigmentosa, and other sort of retinal diseases. Currently, in my postdoc work, I'm focused on degenerative eye disease, another disease that affects the retina, and especially those cells that connect the retina to the brain, which is called retinal ganglion cells. So these cells like transmit all the electrical signals that transmitted from the photoreceptors into back into the brain. So in this eye disease, usually there is like increase in the intraocular pressure or the pressure inside the eye that damage those cells. And once those, like, it's a gradual disease, it builds over time, and there is so many risk factors for it. So once these cells are lost, they are lost forever, because those stem cells that I talked to you about in the previous slide, they won't regenerate. Like after birth, they go into like a state of sleep. They don't divide anymore. They don't produce new cells. So once those retinal ganglion cells are lost due to that disease, they won't regenerate, and they need to be replaced, or patient like receive certain treatment to keep those living cells as they are, without progressing and deteriorating of disease. Because once this disease like progress, patient would have like a tunnel vision where they lost the periphery vision, and just will keep those, like, can see those in the middle of their eyesight or eye field. So to replace those cells, I'm using also a 3D model called retinal organoid that also Kristen has explained. They resemble the human retina in lamination and the function and everything. So I'm trying to develop a protocol to enrich for this specific type of retinal cells, which is the retinal ganglion cells. I mean, now, like, in a stage of examining different protocols, trying to like do several experiments, try different factors that can keep those cells alive and make them more of them. Beside doing the lab work and trying to answer those questions to find answers, I also, like, during lab time, I attend if there is any like lecture lab meetings. Like, we have weekly lab meetings. We have also like research days, departmental social events, lab socials, seminars, lectures, traveling to conferences, and just to get more knowledge of what's happening and occurring in the field. Aside of all that science life, I also have side hustle. I like to, as I told you, to use crochet, knitting to create those models, educate other people about it, host so many events to teach people how to crochet those, like this, for example, the sprain, and like hosting events, going to workshops, to libraries, anything that has like a science, art kind of stuff, events within Toronto. I always like try to go. And also I like to cook. I like to explore the city, walk in the nature, and take these beautiful pictures, because I like also to, as I told you, photography. So for all of that, like loving the science and loving like photography, crochet, science communication, I want to give people like blind, all behind all the things that I'm doing is giving those blind people hope that they will be able to see this world again. Like I'm trying to do all my work because I like to understand the cellular mechanism behind those diseases. Like during my medical school, I was always fascinated how these diseases like occur at cellular level. So now that I have this skills and this position, I can go and understand and do experiment to see the connection between the basic science and the clinical practice. Like it's kind of called a term translational medicine, using those like to educate people about stem cells, stem cell therapy, debunk misinformation, because there is so many misinformation about using stem cells in treatment and in regenerative medicine. Yeah, so I hope like if you have any questions later on to come ahead and ask me if you have anything. I will be really happy to answer any question you have.
Thank you so much, Tahani. Such cool work and such a cool hobby also. Like two amazing things in one.
Okay,
So
thank you.
Tina, you're up next. So just take over screen share when you're ready.
Okay, hi everybody. Nice to meet you all, and great to hear about all my colleagues' amazing work and also to share some of what I do. And wonderful to see so many of you interested in about learning about ophthalmology, the eye, and research and science. So I'm Tina Felfeli. I'm a resident doctor in the Department of Ophthalmology and Vision Sciences at University of Toronto. I also recently finished my PhD in clinical epidemiology at the Institute of Health Policy Management and Evaluation, which is a branch of Daladana School of Public Health, also at University of Toronto. I'm part of the Clinician Investigator Program, also known as CIP, or the Clinician Scientist Training Program, which means that as a part of my training, I'm preparing to be a clinician scientist and also an ophthalmologist. So a little bit of background about me. I grew up in Richmond Hill, Ontario, after I moved to Canada from Iran, so I'm Persian. And when I first moved here, I didn't have any friends. I didn't know anything about the language or Canada. And art, dance, and swimming were a big part of me getting adjusted to this new country and also meeting a lot of new friends. And that was really fun, and it's some of my fond memories from childhood. From there on, after high school, I went on to start to volunteer at a few places throughout my high school training. And at that time, I realized that I really liked the sciences, especially biology was a particular topic that was really interesting to me. And so I knew by the end of high school, by about grade 11, grade 12, that I wanted to pursue a degree in science. So I applied to a Bachelor of Science degree. And from there on, I went on to volunteer at various nursing homes and etc., and became really interested in medicine and sciences. So over the years, I'll talk a little bit more about my education, but I've really been lucky throughout my training to have been able to publish a book. So I've been able to publish a textbook as an author. I've been invited to be an editor on several chapters and other books that have also been published, and also publish a lot of papers that are now posted in peer-reviewed journals and read by people internationally. And it's something that is really meaningful, that you're able to share your work and your research findings with people, not only in your own institution, but nationally and internationally. And so going back to my training, after starting my Bachelor of Science training at Western University in London, Ontario, I started to do more volunteering, became interested in medicine, and applied to medical school at University of Toronto. At that time in medical school at University of Toronto, I wasn't really sure what subspecialty within medicine I wanted to do. I just knew that I first and foremost wanted to be a clinician and always had an interest in research, had considered applying to an MD PhD program initially, but because I didn't know what subspecialty of medicine I wanted to go into, and I wanted my research and my medical degree to be complementary to each other, I decided to defer my PhD training until later. By the end of medical school, I became exposed to ophthalmology, and I'll talk a little bit more about why I fell in love with ophthalmology. But ophthalmology was something that I really wanted to do. I wanted to be an eye doctor where I treat and manage both medical and surgical diseases involving the eye. And from there on, applied to a residency program at University of Toronto for ophthalmology, and was very lucky to be accepted into the program. A residency program is for doctors who graduated from medical school. They're able to now train to subspecialize to be even further specialized in a particular area. So some people may choose to be skin doctors. Some people may choose to be eye doctors, heart doctors, etc. And I wanted to focus on the eyes. The training for ophthalmology subspecialty training is 5 years. Your first 3 to 4 years are your medical training. So you focus on treating any medical conditions involving the eye. And then your last couple of years of training are focused on your surgical training. So that's when you really start to operate each and every day, do various surgeries that you may have heard of, such as cataract surgery or other surgeries involving the back of the eye, such as a pars plana vitrectomy. And it's a very rewarding surgical specialty. From there on, some people may choose, after you graduate from residency, after the 5 years, to go on and to do further subspecialized training, focusing on particular parts of the eye. So within the eye, we have specific areas as well, including the anterior segment of the eye or the front part of the eye. And in the back of the eye, we have the lens. We have subspecialists that focus on the pressure in the eye called glaucoma specialists, people that focus on inflammation within the eye that are known uveitis specialists, people that focus on a particular age group, such as pediatric ophthalmologists. We have comprehensive ophthalmologists, oculoplastic surgeon that focus on the orbit around the eye and eyelids. And so there's a number of subspecialty trainings even within ophthalmology itself. And some may choose to do specialized training even after their residency, which may be anywhere from 1 to 2 or even more years after your residency is done. So currently, I'm in my 2nd year of residency at U of T for ophthalmology. And at the time of starting my residency, I decided that this was the perfect time for me to also do a PhD. And in communication with people in my program, I made the decision to apply to a PhD program. And that's when I started my PhD. I did my PhD for 3 years, graduated, and then returned full-time back to clinical work. And that's what I'm doing now, is my clinical work full-time, which has been very rewarding. So I'll tell you a little bit about my research. So my first research involves looking at cytokine biomarkers. And this is looking at individualized way of treating patients. So how we can personalize management for patients. Currently, the way we treat patients is based on guidelines. So these are generalized guidelines that are available based on evidence that's currently available, usually depending on what's been published in the literature, based on large population of patients or smaller case series. And so there's an algorithm you follow when a patient presents with a diagnosis in terms of what treatments you're going to start the patient on. And these are typically based on what has worked in the past for majority of patients. But not all patients are the same. And ideally, we can actually customize treatments for patients based on their unique diagnosis and their unique characteristics. So we have the advantage now to be able to, using a small needle, take some samples, fluid from the front part of the eye, send those to the lab to be analyzed, and identify inflammatory biomarkers. These are small molecules that are indicative of inflammation and unique characteristics of the patient in their eye and their diagnosis, and can change with disease. We can analyze those in a lab and then use machine learning or AI to find patterns that are unique to that patient So I'm presenting here on the side, you'll see this is hierarchical clustering using AI to identify patterns of how the biomarkers are different in a particular patient. And based on that, we can target the treatment that is most suitable to that patient and not just based on generalized algorithms. So this is the future of how we're going to treat and manage patients, but also monitor their prognosis, monitor how they're actually responding to treatment, so we can intervene faster and make sure the patient outcomes are better because we're able to optimize their treatment. Now, this is another type of research that I do that I'm really interested in. The next one I'll talk about is developing new surgical techniques. So as an ophthalmologist, there's a number of surgeries that we do. Some of the surgeries involve the back part of the eye. You've already been introduced by my colleagues to the retina, which is responsible for your vision. And in particular, macula is the central part of the retina that is responsible for your 20/20 vision. And so this part of the retina is super important. And unfortunately, some patients will develop a defect in the central part of their vision, the macula. And the ways we treat this defect called a macular hole is through surgery. Currently, there's different ways of filling in this defect. Sometimes people will just do a surgery to try to bring that tissue together, to bring that gap together and close that gap. Sometimes the gap is so big that we can't close it. We need other tissue to try to close it. In certain cases, we actually use amniotic membrane, which is the tissue that's been donated by the placenta of the babies that have been delivered from the parents. And this is all stored in a biobank, and it's available for purposes of treating other patients. And this tissue is then put into the eye and fills in that gap that remains open to try to close it. And in most cases, it works really, really well. So some of these really new surgical techniques are allowing us to treat patients better and allowing patients to see in a way that we weren't able to do before. Some of my research now focuses on developing tools to do some of these surgeries, how we can cut this tissue better, how we can insert this tissue into the defect better, and how do we monitor patients' respond to some of these interventions better. Lastly, I'll talk about another aspect of research that I'm really interested in, and that's looking at the population of patients and the overall well-being from a public health perspective. So my PhD was in clinical epidemiology, and as a clinical epidemiologist, I have an interest in biostatistics. So some of that biomarker research that I was doing is translational work involving machine learning and biostatistics. I'm also interested in improving patient outcomes, so that's some of that surgical technique. And lastly, looking at the entire population from a public health perspective. And so this research is focused on diabetic retinopathy. So diabetes is very common. Many of the patients that I see in clinic every day have diabetic retinopathy. And if we can detect and diagnose and treat these patients earlier on, we can prevent blindness in these patients. And so you'll see a map at the top there, and this shows all the patients with diabetes living in our Greater Toronto area who don't have an eye exam to detect diabetic retinopathy. So that means they could be living with diabetic retinopathy, and by the time they realize that they have any symptoms, it may be too late, and some of the vision loss may be irreversible. So we want to intervene as soon as possible. And how can we do that when there's such a large population of patients and not enough doctors or eye doctors to be able to screen those patients? Well, we started doing a screening program where we would go to various community centers, we'd go to various nursing homes, and using portable devices, some of which you'll see on the screen here. These are handheld cameras where we now have new advanced technology. We can take images of patients' eyes to be able to tell right off the bat, if these patients have diabetic retinopathy by taking photos of their eyes. It's a non-invasive way and a very fast and effective way of detecting diabetic retinopathy. And if these patients have diabetic retinopathy, then we can offer them treatments and save their vision. And what's really interesting is now, because there are so many patients with diabetic retinopathy and diabetes that need to be screened for diabetic retinopathy, we can actually use AI softwares that can automate the process of screening these patients. So once these images are captured, anybody who's not necessarily an eye doctor can use these cameras that are portable, even their nursing team, any of their social workers that are able to help and take these images can upload these images onto a software, and the software will automatically tell the care provider whether the patient has diabetic retinopathy or not, and the patient can be provided that information right away. So that's really exciting. It's an area of research that I'm doing now on how to implement some of these services and make eye screening more accessible to patients everywhere. Now, a typical day in my life. So I promised that I would talk about why I fell in love with ophthalmology. Well, a typical day in ophthalmology starts with an early morning. So typically I'll get up at around 6:37 in the morning. And that's usually because that's when I'm on the pager. So you have a specific phone related to your work that patients can call you on anytime throughout the day and night, or any other doctors in the hospital that are worried about a patient with an eye condition, they can contact you at any point. So that's when I usually get a call on my phone about a patient. And then I have to triage or decide how urgently I need to see that patient, whether that patient needs to come to the clinic, or if I have to go to the hospital right away, wherever that patient is located, and see that patient immediately. I then, after start my clinic day, so usually around 7:30, I have patients booked who will come to the clinic, and I'll see those patients throughout the day. And throughout the day, I may be getting calls about other patients, so I may have to step away and come back. If there's any procedures, you'll see me there doing some procedures where I may have to do a laser procedure for a patient in the office, or may have to take the patient to the operating room with the help of my colleagues to do surgeries on the patients. And that will usually take you till the end of the day. And what I really love about ophthalmology is that it's very fast paced. So you're seeing a lot of patients throughout your day. The volume is really, really high. The pathology is very interesting because it's all visual. So you're always relying on your visual cues and what you're seeing on examination to decide what the patient has. And we use a lot of technology, a lot of cool devices, imaging devices, lasers, a lot of really neat machines to be able to diagnose and manage patients. And I find that really interesting. And also the surgery, the microsurgery that we do under microscopes is very precise, is very detail oriented. And I find that very fascinating and intriguing. And it's something to really, it takes a lot of effort to learn about. It takes a lot of effort to get really good at. But once you are able to do it, you really do see the difference and the impact that you're making in your patient's vision. And I always remember the patients where are not able to see, and they're haven't seen their family members for several years. They have a lot of difficulty driving at nighttime, and we're able to do cataract surgery for them, which takes about 15 to 20 minutes, or repair their retina if they have a retinal detachment, which can take about 45 minutes to an hour to do. And after the surgery, the patients end up doing so well, and they're once again able to enjoy their life and have a much improved quality of life. And that impact is fairly immediate. And so you get that immediate gratification as a surgeon and as an eye care provider right away. So that's really why I fell in love with ophthalmology. After a day is over, so usually typically about 6:30 or 7, you then sometimes may be on call. So you see your patients overnight, all night. You may be getting emergency calls about patients, and those nights can get quite busy. Nights that I'm not on call, I spend a lot of time studying. So I'm still a resident. I'm studying in ophthalmology, subspecializing in this area. So I do spend a lot of my time studying. We have a lot of frequent exams. There's a number of new studies and investigations that come up that we have to know about and be up to date on. So it's always lifelong learning, even beyond residency and fellowship. And then research is a big part of my life as well. So even though I just finished my PhD, I still do a lot of research and love to stay up to date with my research as well. And so a lot of the other free time that I have is also devoted to doing research as well. And then, as my colleagues mentioned, conferences are a big part. So traveling to various parts of the world, across Canada, meeting your colleagues, presenting the work that you're doing, but also learning about their work and how that can impact your practice, but also showcasing what they can do to change their practice is really wonderful. And teaching other people. So I'm learning, but it's also great to teach other people who are more junior to me. So I spend a lot of time teaching medical students and also teaching other residents who are junior to me, supervising them and passing on the knowledge that I have gained. I'm also a mom, so taking care of my son, spending time with my husband and my son is a big part of my life as well. And although life is really busy as a clinician scientist and as a resident, that's a part of my life that I always make time for. So every opportunity that I get to see my son and spend time with him, and also my friends, is a really important part of my life. And those are really the people that keep you sane and keep you motivated and keep you going. In addition to your patients who always keep you happy and motivated, having your colleagues around and your family around is really important.
Thank you so much, Tina. Always good to hear that a work-life balance is possible. So what we're going to do now is get started with the Q&A portion of our session. So if the speakers come back on camera, we're going to get started. So the first question I have was just a general, like, why vision sciences in the first place? I guess a lot of kids in high school are like, why would this be your career path? How did you end up randomly in this field? I know some of you covered it in your slides, but maybe we can touch back on it. And Kristen, why don't we start with you because you spoke first?
Sure. So that's a really good question. And sometimes as a grad student, you stumble upon the specific field that you're in a very serendipitous way. So I didn't have a specific field of medicine that I was particularly interested in when I was exploring options for grad school. At the end of my undergrad, I was kind of open to a broad range of things, but I was really, really interested in regenerative medicine. And I had a grandfather who was impacted by glaucoma. So there was a little bit of background there in terms of my interests in retinal disease. So that pushed me to looking at regenerative medicine in combination with the retina and how the two things can get combined. And I found this really amazing clinician scientist who's also an excellent role model for me as someone who would like to partake in that kind of career one day, in my current supervisor, Dr. Brian Valios. And so it all came together and unfolded like that. And I also knew that at this stage, I wanted to do basic science research, which means that you're working at a bench and you're doing the pipetting yourself. You're doing all the cell culture. And so that was heavily a part of the project that I would be undertaking when I signed up for the master's, and it all fit together perfectly because of that. And the I, in my opinion, now that I've been able to be involved in research for 2 years, I think it's like, and we're biased, but it's such a cool part of the body. It's like it's one of our 5 senses and probably the most interesting sense of the 5 in its intricacies and how it allows us to see the world. It's like so cool how we're able to take and capture the entire world in this tiny little tissue at the back of our eye. It's amazing, and send those signals to our brain. So I'm sure Tina and Tahani have more to say on that, but I'll let them speak now.
Yeah, it's like you don't know how cool it is until you start studying it. So
yeah.
Tahani, how about you? How did you get into vision sciences?
So as I told you guys, like it's my hobby motivated me to look for a vision science program. Since I was in high school, I always like art. So I have been always like a member of art classes, art club in the school. I like to draw like traditional pencil drawing, oil color, watercolors. So I have been always like thinking about how beautiful these drawings, how beautiful the colors. Why can we see colors and other people can't see those colors? And then like when I just graduated from high school and joined the medical school, like I started to be interested more like a side hobby in photography, taking pictures, looking at those details. Why can we see these details and other people can't? And as a physician at that time, it was like hard for me to to do a basic research and understand the concept behind my questions. So that's why, like, once I graduated from medical school, did my medical training, I wanted to go back and study why those things happen to some people and other people not. So that was like a motivation for me to try and, you know, contact supervisors, professors who are interested in vision science, try to find a position so that I can, you know, pursue my passion and answer my questions.
It's a fun journey you've been on. Tina, what about you?
I think for me, it was just choosing what I'm passionate about. And I think if you had asked me when I was in high school that I was going to be a clinician scientist and be in ophthalmology, I would have never guessed that. I would have never said that that was something I was going to do. And I think it was just a process of being open-minded, never closing any doors, always saying yes to any opportunities that came my way, and being truly honest and genuine with what I wasn't interested in and what I was good at. And I think that just came naturally. So when I liked research, I pursued it more. When I liked science, I pursued it more. And one step led to another. And I think following your passion and what you're good at and what you enjoy doing and what you're genuine about really gets you to what's best for you. And I think that's really why I ended up doing what I'm doing today. But I would have never guessed that many years ago when I was in high school or when I was in undergrad. So I think doing that, that's probably the biggest advice I would give, with being open-minded and really following your passions, and that'll take you to the right direction for you.
So you're saying they don't have to know their future right now.
I didn't know for sure. So
all right, we got more questions from the chat. Okay, I got a question about, I guess it's more, it could be Tahani and Kristen. They want to know more about the instruments and the tech that you use in your research and how that is advancing right now, what's been changing in the research field of tech. So maybe Tahani, you want to talk a little about the tech you're using?
Yeah, sure. So basically, we are trying to enrich for those cells. So every cell type in the retina has their own marker. So the markers are like kind of receptors that express on that cell that you can identify that cell from the other cell. So when I grow my cells within that 3D organoid, I wanna just like look if I enriched for that cell type. So to do that, I do like a lot of checking those markers using PCR machine, which is like you take the protein or the, you know, the factor that you want, and you try to make many copies of it so that you can identify that these cells express that marker and they are like still alive in your culture, they are enriching that. And also like try to stain those cells, like mark them with different colors and see if those cells are like present in your organoids or in your petri dish. And with advanced technology also you can screen for like the DNA, the RNA in those cells. Like it's called like RNA sequencing. So you sequence those cells to check their RNA and other markers too. So there's so many advancement in that field that can also Kristen talk to you about.
Yeah, Kristen, what about tech for you? Someone even asked, do you use AI?
Ooh. Okay, well, Tahani gave a really good overview of some of the techniques I use. So histology in general, if you guys are familiar with that broad term, basically analyzing tissue under a microscope on a microscope slide. And there's different ways that you can look at the tissue and do different histological analyses. One of them uses fluorescent labels that we put on the slide, and kind of similar to what Tahani was saying, based on the cell type, you can see the different fluorescence that comes out of it, and you can use that to analyze the tissue. PCR is a big one to look at gene expression profiling, which also Tahani talked about a little bit. Something in our lab that we're really excited about is the use of CRISPR, Cas9 gene editing. We're making a new cell line to use as our healthy control cell line to differentiate and derive healthy retinal organoids to compare to the patient organoids. That the only difference between those cells and the ones that we're using from the patients is the gene correction. And to do that, we're using CRISPR, which is that famous technique that was birthed in 2013 or something, Nobel Prize winning technology that we get to now benefit from in our lab. And it's very, very neat. Other techniques, well, just microscopy in general, using these we have the ability to use these $1 million microscopes, which is fantastic. And they're shared between different labs in the facility that give like this amazing image of the tissue that we want to look at the microscopic level. So it's super, super high definition at like a minuscule magnification. Or a huge magnification for a minuscule thing. Those are the big ones. AI, I don't use yet, but that would be something in the future that I'm sure all researchers, clinical or basic science, will integrate into their daily practice. I'm totally sure of that, and I need to familiarize myself with AI for sure.
Slowly coming.
Yeah, slowly coming.
All right, Tina, we got some questions for you. First question is, what is the most difficult part of your job? Do you have a work-life balance?
I think the most difficult is a work-life balance, to be honest. I think as a clinician scientist, anyone who's at this stage, and my colleagues can attest to this, you get to hear by really being truly dedicated to your work and enjoying what you do. And it's hard to kind of draw the line between working all the time because you love what you do and also balancing that with all your other commitments. So if you do arts, if you do knitting or other things outside of work, you have a family, making time for that. And I would say, honestly, the biggest challenge is just not enough time in the day. And there's always opportunities coming up to write a new paper, do a new project. And there's so many things that you always want to be a part of. And at the same time, you have to make time for your family and other things in your life that are important. And so I would say that's probably the most challenging part. I think interacting with my patients and seeing them do well is always really, really rewarding and makes me so happy. Sometimes seeing patients who don't do so well is also very challenging. But that gives me more motivation to do the type of research that we do, to do the basic science research that my colleagues are doing, or the clinical epidemiological research that we do to try to target and identify ways that we can get better. So I actually always see it as a positive way to use some of those areas where we're weak to motivate us to try to get better and improve healthcare.
Another follow-up question for you then. They're saying, you've done a lot of schooling. Is there a specific university or school you would recommend for vision sciences, ophthalmology, all of this stuff?
I don't think so. I don't think there is a cookie cutter formula for schools that you should go to. I don't think there is one perfect school for anything, to be honest. I think people who go into medical school, people who go into visual sciences, all come from various backgrounds, various knowledge bases, various interests. And I think that's actually that diversity of where people come from, what their background is, what actually enriches our program, enriches medical school. In medical school, I came from a typical background of science, but some of my colleagues had a law background, they had an arts background, they had a music background, and those were some of my best friends because they had a completely different perspective on everything that I was learning about. And I loved chatting with them and kind of picking their brain on how they perceive things and what they kind of view things in. And I thought that was fantastic. So I think picking schools is really based on what works best for you geographically, academically, and what your interests are. And you don't necessarily have to go into science if you want to do medicine.
You can do whatever you want, basically. All right, more questions. We have a question about, okay, there's people that are interested in vision sciences, but how would they know if they actually like it? So, how should they get involved in the vision sciences field? Any suggestions or things they can start working on right now while they're still in high school or early undergrad? Kristen, how about you? Any thoughts on how they can jump into vision right now?
Okay. Well, if you're interested in research, as a high school student, there's some labs that I know if you are really motivated and really willing to jump right into the research, you might be lucky to score a supervisor at a research institution that will take you on as a volunteer and be able to shadow the grad students in the work that they do. So being able to just be exposed, maybe not so much the hands-on things, because that's sometimes tricky to get a hold of a position like that in high school, but even just to be exposed and see what it's like to have a grad student life in the lab, see what a pipette looks like, that kind of thing. I think that's a good starting point. That's not specific to vision science, that's specific to researching. Probably Tina can talk a little bit better about being specific towards ophthalmology. So maybe I should pass it to her for that.
I completely agree with Kristen. I think exposure to various fields or people that you see are really, really important, just so that you can understand what fits for you or what are things that you don't like. And starting to understand that earlier and being open-minded to trying things is huge. And then finding mentors, I think that's really, really important. I think the reason why I'm actually here today is because I saw people that I wanted to be like, and I really connected with. They guided me through the way. They also gave me opportunities. I asked for opportunities. And so I think you want to find those people and then get early exposure. I think those are key.
They're great pieces of advice. So I guess, Tahani, you haven't given us a piece of advice. Maybe you can be the last person to speak for the panel. Any piece of advice on these students in high school, thinking about their career, getting anxious, all of these things?
So my advice, it's a social media era, and there is so many great scientists, vision scientists are there out sharing their science, their daily life with everyone. Just try to use hashtags or, vision science, and try to follow those accounts. Look for them, follow those accounts, interact with them. On YouTube, on social media, on Twitter, on Instagram, there is so many accounts that really can benefit all the students who are interested in vision science. You will learn a lot from them, basic science, advanced science, clinical science. Also, you're gonna build your network, so they can introduce you to other people also interested in vision science. And once, you need to start building network right now at this age, at high school age. Yeah, so good luck for everyone.
Yes, networking is important, sadly, but it is. All right, so I'm just gonna do a couple of wrap-up slides. While I'm doing that, Trisha, can you launch your exit poll? We want to know if you've learned something. I hope you have, because I have, but we will have you fill that out. So thank you again for coming to one of our UHN STEM Pathways events. Again, we are this outreach program at the University Health Network. We do these virtual events as well as some in-person events. If you ever want to come to UHN, speak to some of these people in person. So you can check out our website to learn more. You can always connect with us on social media, as Tahani was mentioning. We do have a Twitter account, but we also have a LinkedIn account as well.